All right, it's time for me to start talking. I'm sorry. We got some things to go through today. So let's see, are there any announcements? No, there aren't any announcements. Everything's going smoothly in the lab. I'm just kind of impressed. You guys just cruising right along. I'm looking forward to seeing some data later this week or early next week. It'll all be good. In lecture stuff, yeah, I will post another problem set tomorrow that'll be due on Sunday, and then you've also got another assignment piled on top of that, which is you're going to write the first draft of the chunk of your introduction for your lab report. So I'm just piling the work on you guys. Isn't that nice? So today what we're going to do is I thought this would be a good time to go through some problems. We'll just work through a set of genetics problems that involve dihybrid crosses. I think I threw in a trihybrid cross in here. Yeah, I did. Don't be scared, they're all easy. And we're also looking at this phenomenon, which is there are factors that affect the frequency of the progeny of the cross that don't fit the old Mendelian paradigm. So they're a little different. So this is one example we talked about last time. So this is the Agouti yellow mouse. So yellow mice, we're looking at here, they are going to be heterozygous for the yellow allele. That's what we see up there, A superscript Y. That's the yellow allele. And then they've got the wild type big A allele there. And when you cross the heterozygotes, you get this curious result where it's 2/3 yellow and 1/3 agouti. And the reason for that is if you become homozygous yellow, like this one right here, you are a dead mouse. You don't even make it to term. So instead of the usual 3-to-1 ratios that we see in this again, this is a monohybrid cross. We expect 3-to-1 ratios, right? You don't always get them. We get a 2-to-1 ratio, 2/3 to 1/3. So this is an example of a recessive lethal allele. There are also dominant lethal alleles, and we mentioned one earlier, Huntington disease. So Huntington is a dominant lethal allele, but it has late onset. So yellow, homozygous yellow, kills them in utero, so you don't even see them. Huntington disease kills you when you're middle-aged, so we do see them. All right, so the complication here is we just mentioned this one really simple monohybrid cross that produces a non-3-to-1 ratio, produces a 2-to-1 ratio. What about dihybrid crosses? We can start mixing and matching these different kinds of alleles in a single cross. So here, for example, we're saying, okay, if you get dihybrid crosses, one or more of the traits is not inherited in a simple Mendelian fashion, you won't get a 9:3 to 3-to-1 ratio. You're not surprised by this, right? If you do a monohybrid, you don't get a 3-to-1 ratio. You're going to get some other ratio if you do a dihybrid cross. The easiest way to solve for these is just use the fork line method. And I will hammer on this again. These problems, I could make them incredibly hairy. I could invent such elaborate, complicated, multi-part questions. But you shouldn't be intimidated by them, because what you always do with these, you just split them down into single loci. And you just ask, what does this trait do? How is this other trait inherited? And you multiply the results together. So never be scared when you see a complicated looking problem in this. Well, maybe later you'll be scared, but now don't be scared. they're all reducible to single, simple Mendelian or non-Mendelian crosses. So here's a problem. What are the probabilities of the phenotypes that could be produced in a marriage of two people, both of blood type AB, and both are carriers for albinism? So remember, albino is a recessive trait. So we're again just going to be doing a dihybrid cross. The two loci we're concerned about are the ABO blood group locus and this pigmentation locus. But again, you just take them apart piece by piece, and it will fall out fairly easily. So here we go. This is from your textbook. It just shows you how we do this. So again, you just split them up into the two loci. So here, over here, we just look at pigmentation alone. So we just said that the parents are heterozygous for albinism. So they're carriers. And when they have offspring, 3/4 of them are going to be pigmented, 1/4 will be albino, right? You all know that. That one you should just, poof, comes right out of your head. You don't even have to think anymore to do that. So we're going to get a 3-to-1 ratio here. In the blood groups, so set this aside. Just put a pin in that for 1 minute. We'll go over here and think about just the blood groups. So we got two heterozygotes. We got type A and type B genotypes. And that means that in the cross, 1/4 will be type A, 1/4 will be type B, and 2/4 or a half will be type AB. So the hard part's all done, right? It's easy at this point, because then you just multiply everything together. So if you want to say, how many are going to be pigmented in type AB? You would say, okay, 3/4 times 1/2. So that's going to be 3/8 are going to be pigmented, and type AB. So we can just go through here methodically and get all these combinations using the 4th-line method. And there's our answer, right there. Again, the whole thing looks a little bit complicated, and I appreciate the fact that if I give this kind of thing to you on an exam, it might take a little longer because you've got to work through all the different possibilities. But mainly it's make work. It's going to be mundane and easy. I'll show you some a little later that are a little bit trickier. Okay, so we got that one. Here's another one. So we've got two people. They both have blood type AB. Hey, that's just like the previous problem, right? But now instead of albinism, we're saying they're carriers of the Bombay phenotype. Who remembers what the Bombay phenotype is? Go ahead.
That was where neither the A nor B could be attached because there's something missing for him?
Yes. So you're just gonna, if you're homozygous for the Bombay phenotype, it means that phenotypically you will appear type O, and it doesn't matter what the other loci say. Okay. So we've got individuals who are heterozygous for the blood type, heterozygous for the Bombay phenotype, which is a little messier problem. Okay, once again, we just split it in two. So first, here's the blood types. Just think about ABO. And this is the same result we got before. So we know 1/4 are going to be type A, 2/4, 1/2 are going to be type AB, and 1/4 are going to be type B. All right. Then over here we think about the Bombay genotype. So they're heterozygous for little h. And what that means is 3/4 of them are going to form that H substance, which will allow the other blood antigens to be expressed. And 1/4 will not make it, which means no matter what other antigens it could make, they won't see them. It will be obscured. Now, when we sit down, we're just going to multiply those all together. So here you see we got the blood types over here. 1/4 type A, 1/2 type AB, 1/4 type B. And then we got 3/4 of those are going to form the H substance, and they will express that genotype. So they will be blood type A. But 1/4 of them are not going to have the H substance, so they won't express this. They've got the enzymes that would just love to attach those glycoproteins, those carbohydrates onto the glycoprotein, but they can't. So they will be type O. So 1/4 times 1/4, 1/16 type O, 3/16 type A. And you just do the same thing for each of these. Just multiply them out. You get these ratios. The key significant observation here is that you get 1/16 type O plus 2/16 type O plus 1/16. So 1/4 of the offspring of these two AB parents will have blood type O. And everyone sees that, right? Yeah. See, it's not tricky at all. I emphasize this every year. These problems, you just have to take them apart piece by piece, and then the answers just kind of fall out. So here's another one. So what we got here is here's a new allele I'm mentioning, prop 1, that leads to dwarfism in mice. And it's recessive. And then I tell you this is the problem I give you. And again, don't be intimidated. Sometimes it sounds like I'm speaking gibberish here. But the question is, what are the ratios of the phenotypes of the progeny of a cross between a yellow dwarf mouse and a yellow prop 1 minus prop 1 mouse? So we got a yellow dwarf. So that's going to be heterozygous, right? And we got the dwarf mouse. That's going to be prop 1 minus prop 1 minus. And then we tell you over here we got another heterozygote here. Now we want to figure out those proportions. And one of the first things you should always do is when you see a problem with all this genetics gibberish, all these comfortable genetics words, break it down. What is the genotype of this cross? If you had to write it out in notation somewhere, just the genotypes of the parents. What are the genotypes of the parents? So for instance, this one here, the yellow dwarf mouse, what is its genotype going to be?
Is yellow dominant or?
Yellow is, it's complicated. Remember, that's the lethal one?
Yeah.
So if you're only going to be yellow if you're heterozygous for yellow.
For the first one, would it be they're heterozygous for yellow, and then
And this one is recessive, remember?
and then prop 1 minus prop 1 minus?
Yes. Yes, and often that's the first step to solving these problems is figure out the genotypes of the parents. Okay, so that's one. What about this one? Look at there, I've given you half the genotype right there. What's the other half? It's yellow.
Heterozygous for yellow and
Yeah.
prop 1 plus.
Okay. Yeah, so there's our genotypes. And I find it easier to get it into this kind of phrasing, so it's kind of mechanical than what I have to do. Okay, so I want to know the results of that cross. So let's just look first at a goodie. This one, the yellow allele there. So we're taking A superscript Y, A plus, so it's wild type for agouti there by AY, A plus. What will the progeny look like? What's the distribution? Just looking at yellow, what's their phenotypes? What are the phenotypes we're going to see? I'm not going to tell you, you got to tell me. Yes?
Is it 2/3 yellow and then 1/3 agouti?
Correct. So we went through that problem earlier. Yeah, so it's a standard lethal allele. We're going to get 2/3 are going to be the heterozygotes and 1/3 are going to be homozygous for the wild type allele. Okay, then we take this one. Prop 1 minus by prop 1 minus, prop 1 plus. What will the distribution of the phenotypes for that be? So here we get a homozygous for the recessive trait, and here's a heterozygote, and we cross those. Simple Mendelian monohybrid cross. Nothing funny about this one, no lethality, nothing like that. Go ahead.
Going to be half hetero and half homozygous?
Yes, so half will be heterozygous, so they'll express the wild type phenotype. That'll be half agouti and half normal sized, and half will be dwarf. And then we just have to multiply those together. So that's what we get here. So there's our 2/3 yellow, 1/3 agouti. I just make a note here, the homozygotes die, you don't see them, so you don't really have to worry about them in this problem. And then half are dwarf, half are wild type. We just multiply those together, get all the combinations. So 2/6 are going to be yellow dwarf, 1/6 are going to be agouti dwarf, 2/6 are going to be yellow wild type, and 1/6 are going to be agouti wild type. Again, a useful cross-check with this kind of problem is add those up. If those don't add up to 1, you did something wrong. So you should always get 100% of the progeny you get. So that should add up to a whole 1 of these 4 different combinations. If it doesn't, that kind of tells you, hey, maybe I missed a combination. No, you didn't miss a combination here. Maybe I multiplied something wrong. All easy checks you can do. Okay, let's do another one. Uh-oh. This is the 3 different loci cross. But you're not scared, right? You know, you just take it apart piece by piece. So I'm saying, okay, we got cucumbers, we got dull fruit is dominant to glossy fruit. Orange fruit is dominant to cream fruit. Bitter cotyledons are dominant over non-bitter cotyledons. And we got a plant homozygous for dull orange fruit and bitter cotyledons crossed with one that has glossy cream fruit and non-bitter cotyledons. Yeah, you just read that smear of English and it just runs right past you, doesn't it? So what you have to do first is, here's their first question, just to figure out the genotypes. What are the genotypes I'm talking about here? Technology. You can never trust it. Okay, anyway, so we want to figure out the genotypes of these individuals. So let's do that. So we were saying, okay, we got a plant that's homozygous for dull orange fruit and bitter cotyledons. And look at there, I've given you abbreviations for all of these. So what is its genotype going to be? Go ahead.
All capital 2 D's, 2 R's, and 2 B's.
Yeah, big D, big R, big B, big B, whatever. So that's easy. And then we say, okay, we're also going to cross this to a plant that has glossy cream fruit and non-bitter cotyledons. Notice, I didn't say it's true breeding. Okay, this first one I said it's homozygous, but here I didn't say it's true breeding, but you can still figure out the genotype, right? So somebody else, what is the genotype of this second parent?
It'll be lowercase.
All, yeah, so all be recessive. And that's how you know it's not heterozygous, because you wouldn't see heterozygotes. Okay, so we figure out the genotypes of the cross, it's big D, R, big R, big R, big B, big B. Yeah, just like you guys said, okay. And we know the progeny then have to be 100% heterozygous at all three loci. We all good so far? All right. And I say, okay, give the phenotypes and their expected proportions in the F2s. Oh boy, okay, we're going to cross this by this. We're going to get these heterozygotes in the F1s. And then we're going to cross big D, big R, big B, little b by big D, little d, big R, little r, big B, little b. So we're going to do the next cross to make the F2s. And here I ask, what are the phenotypes and the expected proportions in the F2s? And the answer is, remember, if we've got a dihybrid cross, we get 9-3 to 3-1. Here it's going to be messier because we've got a trihybrid cross. So how are you going to figure that one out? What do I always tell you about these kinds of problems?
Take it apart.
Take it apart, yeah. So let's just look at them locus by locus. So, for instance, the D locus. What proportion do we expect to see of that in the F2s? So we're just asking for phenotypes.
3-1.
3-1. Yeah, so we expect 3 big D and 1 little d. What about this part, the R's?
3-1.
3-1. Oh man, this is a baby problem when you look at it this way, isn't it? Okay, what about B's?
3-1.
3-1. Oh man, this is a joke, except then what you have to do is you have to multiply all the combinations together. So the frequency of big D, big R, big B is going to be 3/4 times 3/4 times 3/4, which is 27/64. And then you have to go through all the others. And I won't bother to try to do them all here. Here's the final answer though. Yes, there are 1, 2, 3, 4, 5, 6, 7, 8 different combinations that result. And there's their proportions. So I'll leave it as an exercise for you to go through those and see that they all multiply out correctly. Okay. I threw in another part to this question though. And this one I say, okay, we're going to take the F1, this guy right here, and instead of crossing it to another F1, we're going to cross it to glossy cream fruit and non-bitter cotyledons. What's the genotype of this one? So I know we got this genotype crossed with the genotype associated with this, which is all lowercase. Yeah, it's all the recessive alleles. It's the same as this one. So another way I could have said this is I could possibly back cross the F1 to its parent here. And we'll get a result there. Once again, you got to break it down into the 3 different pieces of the puzzle. And what answer do you get? What do you get for just the D's? So crossing big D by little d, little d. What phenotypes will we get? Say what?
You got all big D.
No, big D by little d, little d.
Half and half.
Half and half, yes. So we'll get 50% of each, and we can do the same thing for the other three, and then we multiply them together, and presto, we get this ratio. Equal numbers of each of the 8 possible arrangements of the alleles. Any questions about this? It's really easy for me to lead you by the nose through it, isn't it? But I think once you practice a bit more, I will give you problems to practice on over this weekend. You'll see, this is sometimes a little tedious, but it's not difficult. Okay, now this problem is going to look a little bit familiar because it's what you're doing in the lab. So this is asking about the potential results of a complementation cross. So I'm saying, okay, we got some fly mutants. One you named Curly because it's got curly bristles. So all of its bristles are all nice and curly. And the other you call Peg because its bristles are all short and stumpy. And after breeding them together, you make sure that you got pure breeding crosses. And you're going to cross curly flies with peg flies. This is exactly like what you're doing in the lab. Remember, in the lab you're crossing scarlet by brown. Just substitute in curly and peg. And we're asking what is going to happen. Now in this case I'm saying, how would you interpret the experiment if you get the following results? So this is 1, 2, 3, 4, 5 different possible results. And I'm asking you to interpret them. Okay, so you do this cross, and in the F1s, all of the flies are curly. What does that mean?
Curly gene is dominant.
Curly is dominant. In this case, it also tells you that curly and peg are probably allelic. So they're probably at the same locus. Okay. So that's pretty straightforward. This would be like if you cross scarlet and brown-eyed flies and all of the progeny were scarlet. Which I hope doesn't happen, but we'll see. Okay, conversely, what if you do this same thing, all the flies are peg?
They're at the same locus, but peg is dominant.
Yeah. So you begin to see the utility of doing this kind of cross. You're just kind of going through it and figuring out what the potential results are. What if they're all wild type? They all got long, straight bristles. This is a little more subtle. How would you interpret that result? This is like in your cross in the lab, if you cross scarlet and brown, and you get all wild type colored eyes, what does that tell you about scarlet and brown?
Did you say there are 2 different light shades or blue?
Yeah, that tells you they're different loci. So that they've complemented each other. So yeah, because they complement, you're looking at 2 different loci in this particular kind of result. What if you do this cross, and instead of curly, instead of peg, instead of wild type, you get something new, you get stumpy curled bristles. Little pigtails jutting out of the side of the fly. What does that tell you? How would you interpret that? Did they complement? No, they did not complement, right? So that suggests they're at the same locus, right? But they're neither curly nor peg. What does that say?
Is that codominance?
Could be codominance, yeah. So you've got a different relationship between the alleles there. Okay, what if you do this lovely experiment and then all of the progeny die? You get nothing. You put males and females together and no, they don't. Maybe they lay eggs. Maybe you see little embryos. Maybe they go to their first instar larva, but then they all die off on you. What do you know then? What have you learned? Somebody?
It's a lethal, like, allele, or?
It's a lethal allele, yeah. But have you learned anything besides that?
It's a dominant allele.
Maybe.
Or do you call wild type?
Say what?
Would that be like a wild type situation where, like, I don't know, like she said, they're legal?
Yeah, lethality is not wild type. Lethality, wild type lethal would be just terrible. You wouldn't see any flies. Go ahead.
Belong to the same locus.
Maybe, yeah, they could belong to the same locus. This is actually a really interesting result. But it's a complicated result. You got to do more experiments to resolve this one. There was once upon a time when people just sort of threw up their heads and said, oh, it's all lethal. Throw those flies out. But then some of the best progress in evolution and development has come from people who took this kind of result and said, no, there's interesting things going on there. What's going on? So we have a lot of possibilities here. One is epistatic interactions. That you got two loci, all right, and they are both affecting bristle development. And having the combination is just too much. It just causes all kinds of complications, and the fly dies as an embryo. Just to fill you in a little background here, the genes that are involved in making bristles in flies also have many effects in the nervous system. So maybe you've got an interesting mutant that demolishes the nervous system if you put these two in combination. So that, if it's epistatic, that would suggest we're looking at two loci. But it's also possible that you've just got some interaction between two alleles at one locus that leads to lethality. Sometimes a double mutant at one locus is just too much. Again, think back to that agouti and yellow effect. If you've got two yellow genes, that the animal's not going to survive that. It's lethal. Maybe having curly and peg together is lethal. So this is kind of a complicated result. It means, oh boy, I get to go back in the lab and do some dissections and figure out what's going on and get different alleles of these same and yeah, there's my career for the next 4 years. Okay, here's another one. So we've got more fly mutants we've invented. So you get some fly mutants and you breed them to get a homozygous stock of each, so you get true breeding flies. The two mutants are one that makes orange wings and the other makes green wings. All right, you do some experiments, you figure, okay, orange and green are two different loci. So that's a presupposition here. So they're non-allelic, they're two different loci. But when you cross the flies, all of the F1s have orange wings. Green just disappears. But again, I emphasize, these aren't alleles. They're two different loci. Then you cross the orange ones, and you get either orange or green. And when you cross, that's when you cross the F1s, you get either orange or green in the progeny, and you get this ratio. 9/16 orange and 7/16 green. Whoops. Yeah, you should be thinking to yourself, wait a minute, if this were a monohybrid cross, I should see a 3-to-1 ratio. If it's dihybrid, like I just told you it is, or different loci, you ought to get a 9 to 3 to 3 to 1 ratio. What is this 9 to 7 stuff? How do I explain that? So what are the genotypes? Oh, don't give you the answer yet. What are the genotypes of the F2s? First of all, maybe we should step back a minute and say, how would you logically explain this result? How would you just give a rough cut version of how would you explain this in your head? Or how would you get a 9 to 7 ratio? What a weird combination. Are you raising your hand?
No, I was just thinking, like, would it be a case of, like, the position effect where, like, you have...
Oh, we haven't talked in detail about the position effect. Interesting idea, but no, I'll just exclude that explanation. Maybe later I won't be able to, but here. Any ideas? What's funny about those numbers? Okay, if we were expecting a dihybrid cross, and I said we think we should get a 9 to 3 to 3 to 1 ratio, but we get a 9 to 7. Where could the 7 have come from?
Is it the 3 to 3 to 1?
Say what?
Would it be the, like, if it was the dihybrid 9 to 3 to 3 to 1, would it be the 3 to 3 to 1 would have the same phenotype?
Oh, yeah, there you go. So if we expect a 9 to 3 to 3 to 1 ratio, but we get 9 to 7, one explanation might be that those got all lumped together. Those are all making the same color. So instead of the expected 9 to 3 to 3 to 1 ratio, we get 9 to 7, because those are all consolidated. How would we think of the genotypes for such a result, though? So I'll just go ahead here. Here's one possible explanation. So here I'm saying we're going to call those loci O and G. So big O and big G, little o and little g. And then I'm going to include a little notation here. Asterisk means that it doesn't matter. So if you're orange-winged, you have this genotype. Big G and big O, somewhere in there. And that could be, for instance, big G, big O, big O, big O, little o, etc. All these different combinations. It just doesn't matter. They'll give you orange. Similarly, green means that in one of the loci you are homozygous recessive. So if you're homozygous recessive for little g or little o, you're going to make green wings in this example. Yeah, logic got a little bit twisty here, didn't it? Okay, so down here say, okay, explain it to me in English. Just give me a simple English explanation of what you think might be going in here. So we started to get at it over there. So we're saying that 9/16 are flies that have the dominant wild type allele at both loci. So those combinations, you see there, all of those will give you the green, or this was orange winged, right? I'm getting all, yeah, those are the orange winged ones. And we're saying if either locus lacks the dominant allele, like here or here or here, etc., the default is to make green wings. You have questions? I'm going to give you some more problems like this, so ask me questions. You see how this works? You're just trying to work out which particular combination of alleles gives which color and lumps things together in a way that makes that 9-to-7 ratio. Yes?
I can understand how it works, but why does this work? Why do we do
Oh,
this?
why does this work? Okay. Well, what it could be is, for instance, there could be some kind of epistatic interaction between them. So you got to have a functioning G and you got to have a functioning O. And if you got both of those functioning, you can get this color of orange expressed. If either one is non-functioning, there's a failure of the whole system to produce color in the wings, it lapses to a default of green. So that's one possible explanation. You'd want to do more experiments to figure out exactly how it's working. But that's the logic behind it. Okay, in your textbook, there is this delightful table of non-Mendelian ratios that result from various kinds of situations. So if you look over here on the far right, you sometimes get 9-to-3-to-4 ratios, 12-to-3-to-1. There's the 9-to-7 we just saw, right? And then all these other combinations, 13-to-3, 6-to-3 to 3-to-4. How confusing this all is. Okay, so we got these different possible results coming up. And in all the examples here, we're going to say we start with just two loci that have variance. We have mutants in two different loci. And we want to think about what are the ways we can generate these oddball ratios that you see over here. Now this table, again, is in your book, but I've added something here that I hope will clarify it. So this up here, the 9-to-16 ratio is associated with combinations that have one of the dominant alleles at both loci. So if we're saying we got two genes we're dealing with, A and B, and we look at a dihybrid cross, the 9-16 is the dominant one. That's where you're dominant at A, and dominant phenotype at A, and dominant phenotype at B. And the way you get it was with this combination where the asterisk means don't care. You could be big A, little a, or big A, doesn't make any difference. You're going to have the big A phenotype. Okay? And over here, the 3/16 means you've got a big A at one locus, and we don't care about the other. And we got little b. So you got the dominant recessive arrangement. The other 3/16 is recessive dominant. And the 1/16, you've got recessive. So just visualize that in your head. That will help puzzle out these different ratios. So now I'm going to ask you to explain all of these to me. Can you do it? Okay, I'm just going to ask you for a plain English explanation how you would rationalize this. So, for example, here we got, let's go to the first one. We got agouti, albino, black, albino. So 3/16 albino, 3/16 black, 1/16 albino again. That gives us a total of 9-to-3-to-4. What's going on here? What are the relationships between these loci and these alleles that give that particular combination of colors? See, I'm sitting down. I'm just going to relax. You could explain these little logic problems to me. So how does that work? What's your explanation for that? What did the two loci do? Hypothetically, speculatively, how would you rationalize this result? See, the logic problems get harder and harder over the course of the semester. Let's see if we can figure this one out. If I jump in and do the first one, will you all be brave enough to do the others? Look, we scared that person off right away. Okay, so one way we might explain this first row is, okay, we got the wild type phenotype as a product of two dominant alleles at two loci. Okay? That's all we care about there. If the B locus is recessive, it's going to be albino. And look, that's what's in common here and here. So B, for instance, could be the enzyme that makes the pigment. So that's responsible for just making the whole pigment. And then the A locus, you see it's recessive here, and there we get all black. Remember, agouti is that distribution of pigment along the length of the hair. So maybe A is responsible for distributing that pigment in a particular way. So in this example, the way I would explain this is, okay, the albinos are the product of completely lacking the pigment. There's nothing to distribute. This black variant is the result of making the pigment. The B locus is active, but it's not getting properly distributed by the A locus to make the agouti pattern. Does that sound plausible? Yeah. Again, I'm making some hypotheses there. Specifically that, for instance, B is an enzyme for making the pigment, and that A is somehow regulating the distribution. But then, you see, then I get to go back into the lab and do more experiments and take the whole thing apart. Okay, let's do the next one. Squash. Yeah, you've seen lots of funny looking squash, haven't you? You just go down to Willie's. You always have weird looking squash down there. So we got 12/16 are white, 3/16 are yellow, and 1/16 are green. How do you explain it? Yes?
When the A locus is dominant, you get the wild type. It doesn't matter what B is, but when it is recessive, B is responsible for the pigment, and then that depends on whether or not it is dominant or recessive.
Sounds good to me. Yeah. So then what you could think of is, okay, maybe A is expressing, maybe it's expressing a white pigment gene that's kind of overwhelming everything. So when you got A in the dominant state, that's all you see is white. And if A is recessive, like it is in these two cases, that lets other colors show through. So letting yellow or green show through. So yellow and green might be due to the color, might be produced by the B gene. Again, hypotheses that we will go into the lab and test. Okay, so that gives us that 12 to 3 to 1 ratio. Okay, who wants to do this easy one? This is just like the one we just solved a moment ago. You've got a 9 to 7 ratio. How would you explain that? Be brave. This is the easiest one of the bunch, right? Sure.
For the purple, you need both dominant on the two alleles.
Okay.
And then for white, it's if it has any recessive, it'll be white.
Yeah, so the purple color is the result of a combination of something that is produced by A, presumably. So you need A to get, you need A and B to get the purple color. And if either one of those is missing, the poor pea plant just makes white. So you get 97 there. Okay, here's a fun one. Squash, again. Squash, it's weird stuff. They've got all different shapes of them. You know, you go down around Halloween and they always stock all those weird looking squash plants. So you can get squash that's really squashed and looks like a disc, or you can get squash that's nice and round, or you can get squash that's elongated and thin. Okay, those are all different hybrids here. And the ratio is 9/16 disc-shaped flattened, and 6/16 spherical, and 1/16 really long. How do you explain that? Sure.
To get a disc shape, there has to be a dominant allele present in both A and B. To get a sphere, you only really need one or the other to have a dominant. It doesn't necessarily matter which one it is, just as long as there's one that's there. And long has to be completely recessive.
Right. Yeah, one way I like to think of it is that, you know, there's all those strings and things inside a squash. You can imagine there's two kinds of strings that are pulling from top to bottom and deforming the shape of the squash. And if you've got double strength strings, you get squished down into a disc. If one of those strings is missing, you relax into a sphere. And if no strings are present, it just stretches out into a long shape. Again, more hypotheses. Man, too bad this course isn't a couple of years long. We could do so much. No, you're relieved it's not. Okay, here's a messy one. Chickens. Oh, so we got white and colored and white, and we got a 13:3 ratio. Now, this is kind of like Darwin's observation when he was looking at pigeons, and he said, hey, white, it's strongly inherited, right? Because when he's looking at those crosses of black and white birds, you know, chickens, he saw more white progeny than black. So he said, it seems to be strongly inherited. But here it's a little bit messier than that. So we got these 12/16 are white. Then we add in this 1/16, so 13/16 are white, and it's only this one that's colored. How would you explain that logically? I think the key here is to figure out why are these 3/16 here colored and the others aren't? Yes?
B is only able to express a colored phenotype if it has a dominant allele, but only if A isn't. And then when A does have any sort of dominance, no matter what B is, it overwhelms it with the white color.
Yeah, so this is a logical exclusion of sorts. That, yeah, we can make colored chickens with the B locus, if the B locus is dominant. But if we don't have the B locus, we can't make color, so that's this 1/16 here. And if the A locus is dominant, then it wipes out, it whitewashes the entire bird and you don't see any. So this is the only case where you get colored chickens. Okay, don't let us leave you behind. Are you seeing the logic behind these? We're just trying to work through and come up with reasonable explanations for why you would get these variants from the 9:3 to 3:1 ratio. Okay, here's a fun one. This is mice. Yeah, you've all seen white lab mice. There are also white spotted mice, and there are colored mice. The color means brown in this case. So you get a 10:3 to 3 ratio. How does that work? See someone back here who hasn't spoken up yet. Go ahead. Don't worry, you can't get it wrong because I will correct you and will guide you to a reasonable answer. So it's all safe. Just try to make sure you all stretch your brain a little bit.
So how do you explain this result? It's kind of interesting, isn't it, that if you've got a dominant allele at both loci, you're white spotted. If you've got a recessive allele at both loci, you're white spotted. So what could be going on here? That makes these kind of interesting. Wow, what's causing that? White versus colored. We eliminated the spottiness in these. Don't be shy. These are just fun little brain teasers of the kind I could throw at you on an exam. No pressure at all. So how would you deal with this? Again, okay, go ahead.
It's kind of like codominance. If the two alleles are equal, then both colors will show through. But then if one is dominant and the other isn't, then the dominant one will stay.
Oh, okay, that's interesting. So it could be a balance issue. If A and B are in perfect balance with each other, then you're going to get a spotty coat. Both of those genes are going to show. If it's out of balance, so for instance, if you got lots of A is dominant here, but B is recessive, then you turn out white. If B has got a dominant allele, but A is in the background, then you get a colored coat. That's an interesting idea. Yeah, so it's just, sometimes we'll see that. But genes, it's called gene dosage, and it matters a lot in many processes. We'll talk more about that when we talk about sex linkage. So that could be what's going on there. Yeah, so that's a good A. Okay, you get an A on the exam. Okay, next, shepherd's purse. We got a seed capsule, and we got this weird ratio of 15 to 1. It's almost always triangular, but in this one case, it's ovoid. How do you explain that one?
Any type of dominant allele will lead to a triangular seed.
Uh-huh.
And when there's all recessive, it'll turn into ovoid.
Yeah, so this could be a lovely example of redundancy. That if you're trying to make a triangular seed capsule, you've got two different genes that can do that for you, and you're only going to see them lapse into this ovoid shape if both of them fail on you. So yeah, that's as simple as just redundancy. That's what's going on here. Oh, and then a messy one down here, the flower beetle, and its color. Yeah, I raise flower beetles because they make mealworms that you feed to your spiders, like everyone's got at home, right? No. Anyway, so they turn into these little dark beetles that the spiders can't eat because they got really hard, crunchy cuticles, so they get thrown out. Anyway, so here's the distribution we get. Oh, this is complicated because when you have the dominant allele in both the A and B locus, you get a mix. You get 6/16 sooty and 3/16 red. What could be going on there? And then, of course, here we get black and black. Those just lumped together. And jet, which is really black in this case. So what's the logic behind this distribution? Getting a 6:3 to 3:4 ratio. Maybe we should just look at these two combinations. Black and black. Okay. How do you get black?
Do you have a recessive B allele?
Yeah, so it seems like if you've got two recessive alleles at B, you're just going to be boring old black. Okay. Now, if you've got a recessive A and a dominant B, then you get jet. How do you explain that? What's the genetic logic behind that? Okay, we got to explain this one. So it seems like the case here is, this is the only case where we've got a dominant allele at B and A is recessive. And so we get this dark black up here. This case, we'd have to take apart the genotypes apparently, wouldn't we? It's not enough just to look at the phenotypes. We have to, what's the distribution of genotypes here? And figure those out. Okay, so what I'm going to do is I'm going to stop there and let you simmer on this one. So on Monday, I'm going to put this slide back up and somebody is going to explain to me this last result. What's a logical explanation behind this? Okay, I will let you know on that. So again, more problems over the weekend. Work on those, work on your introduction to the lab, and we'll see you on Monday. I think I might start talking about sex linkage on Monday.